So welcome everyone to our newest episode of Spatial Conversations. My name is Marta Gabrielska. I'm from Millennium Science, distributor of 10x Genomics, which I always say is my favorite supplier. Well, I'm really excited about the topic today. I think it's a really interesting thing to discuss, and so hot. I hope it will warm us up because the weather seems to be, unfortunately, going back to winter, especially in Melbourne. So also it's Friday the 13th, but I think we can definitely do it. So today with me, I have Kristen from 10x Genomics.
So hi everyone, if I haven't met you before, my name's Kristen North and I'm a 10x Channel Business Executive located in Sydney. So a little bit warmer, but not warm, I would say. And it's lovely to be here today. So it's great to see everyone joining, and the idea behind this series is to get a little bit more of background information around how people do their projects and unpublished details about performing spatial experiments. This is kind of a new and fast-moving field and hot, as Marta would like to say, and sometimes it's hard to get the information that you need to decide how best to approach your own experimental needs. So these experiments do require a lot of investment of time and precious budget and precious samples. So this series is really intended to highlight different projects. We've got a lot of great speakers here and their experimental procedures and approaches that they used. And we'll open the floor as well to ask questions that you might not normally get to ask these presenters. So we hope you find it really valuable and encourage you to ask as many questions as you want. We have a long question time at the end. So please pop your questions into the chat and try and make it as conversational as possible. So yeah, thank you.
Yeah, thank you, Kristen. Yeah, just a reminder, please put the questions during the talk to our chat, or you can later also raise your hand and ask your questions directly. So as you can see, we have a very strong female scientist team here today. And with that, I would like to introduce Dr. Saskia Freytag from Whittle and Eliza Hall Institute of Medical Research in Melbourne, that will present her work about integrative spatial multi-omic workflow for enhanced understanding of glioma biology. Over to you, Saskia.
Thank you. I'm just gonna share my slides. Does that work?
Yeah.
So yeah, thank you for the invitation and the kind introduction as well. So I co-lead a lab with Drs. Jim Whittle and Sarah Best at the WEHI studying brain cancer. And we've established the lab nearly 3 years ago. But today I want to give you some insights in how we use spatial multi-omics technologies to really understand glioma biology. So patients diagnosed with brain cancer are really faced with quite startling statistics. So while aggressive research and effective treatments have afforded impressive survival outcomes to previously terminal cancers, such as leukemia and breast cancer, the 5-year survival for brain cancer is only around 20%. So that means that 80% of patients, many of them who are under the age of 40 and have families, will die within 5 years of their diagnosis. So brain cancer is a really heterogeneous group of cancers, and our lab exclusively really focuses on the two most common types, and these are glioblastoma and diffuse astrocytomas. And together they are referred to as glioma sometimes. And these, or at least GBM, happens to also be the one which has the poorest survival. So we have really long known that the extent of surgical resection for these cancers really correlates with the overall survival in glioma. And that suggests that glioma cells infiltrate into the eloquent brain. And indeed, recent studies have confirmed their presence in the eloquent brain, but also that they form synapses with neurons, intriguingly. And these glioma neuron synapses actually promote tumor proliferation and plasticity. However, we are really yet to identify any ways to target these infiltrative tumor cells, and they are often the cause of recurrence in these patients. So it's really paramount that we do so. So spatial technologies are clearly poised to help us to answer this question and understand how these infiltrative glioma cells interact with the normal brain cells. So early spatial transcriptomics technologies that we applied in the lab and others have applied revealed that gliomas such as the most aggressive form, GBM, which I'm showing here, are immensely heterogeneous, even at close proximity. So that means that the tumor cell states are really appear in very different compositions in regions that can be quite close to each other. So using single cell resolution spatial transcriptomics, such as the 10x Xenium, we were able to refine this model even further and show that these different tumor cell states are actually intermixed in the tumor core. So we now want to turn this tool in combination with spatial metabolomics to really study the infiltrative tumor leading edge in low-grade gliomas in order to construct a multi-omic model of what is happening there. So to do this, we collect spatial transcriptomic, metabolomic, immunofluorescence, and conventional histology data from the same block of frozen tumor. And to achieve this, our postdoc Göttingen, who is actually an imaging specialist, had to optimize several techniques to allow us to overlay these very different types of information. And they come largely from sequential slides, which makes this actually quite difficult. And also that we're using frozen tumor pieces, which we do need to do because of spatial metabolomics actually makes this rather challenging as well. So first he had the clever idea to generate a micro CT scan of the frozen tumor piece. And this allows us to direct the cryosectioning in a way that we actually get sections that have the fewest tears or folds. Once this is done, and once the cryosectioning is done, the slide that is destined for Xenium is sent to the WEHI Advanced Genomics facility, where it's analyzed using a custom panel of around 300 genes with the Xenium instrument. And so far, this has really worked quite perfectly for our lab. We have sent about 45 different tissues to the WEHI Advanced Genomics facility, and they have all pretty much worked perfectly. So after the acquisition of the spatial transcriptomics data with the Xenium instrument, we then perform post-staining with several antibodies of interest, and that is to improve our cell segmentation. And I'll go into some detail afterwards as well. So while I have just told you that we obtain nearly all these modalities separately from sequential slides, I do briefly want to mention that you can actually get a surprising amount of data or different techniques out of the same slide. So we have not been able to just do immunofluorescence, but also a conventional H&E after the immunofluorescence on the same slide that was used for Xenium. And you can see the data here, which looks beautiful. The conventional H&E is not quite as stunning as it would be, but it's still good enough to work with. So back to our spatial multi-omics data that I'm sure you can imagine produces a ton of data that requires an actual very complex computational pipeline in order to overlay the information, pre-process, and make it ready for downstream analysis. So the first step in our pipeline, which I will take you through in detail, is to segment cells. And for this, you have several options. So first off, you can use the expansion model that 10x provides, which from the nucleates dilates outwards until it touches the next cell. Second, if you have immunofluorescence available like we had, so we typically stain for GFAP, phalloidin, and DAPI because we are brain samples and brain tumor, in particular GFAP stains for the cancer cells. You can actually train a custom model for identifying those cell boundaries, and we use CellPose in order to do this. And third, you could just simply segment the nuclei only and only use those as your cell masks. We also use CellPose for this, but I just want to say that 10x also provides this natively. So first up, I want to assure you that all the different segmentation strategies identify a decent number of cells. But the immunofluorescent-based segmentation beautifully captures the cellular morphology. And you can see this really here with this astrocyte-like cell that you're seeing where we have zoomed in rather closely. Unsurprisingly, expansion-based segmentation produces cells that are on average 3 times larger than those determined with our custom cell post model. And we found that in particular in densely packed areas, the neighboring cells really strong, or the amount of neighboring cells really strongly determine the cell size in the expansion-based strategy. Immunofluorescent-based cell segmentation is also really great in order to identify multinucleated cells. Well, actually, it's the only way you will be able to identify multinucleated cells. So multinucleated cells are obviously common in cancer, but they also identify cells that are undergoing cell division. In our sample, we found about 5% multinucleated cells. And then when we checked in what cell types they are enriched, which is the picture on the right-hand side that you can see, and these are the ones that are above the line, you can see that these are, in fact, mostly tumor cell types, as well as cells that are dividing. And we don't see many of these multinucleated cells in cell types such as neurons, as you would expect, because these are not typically dividing cells in the adult human brain. So while it is fairly obvious that cell segmentation produces cellular morphologies that are more accurate and also allows for the identification of multinucleated cells, it was really interesting to us that when we looked at the annotation of the cells, either when using cell-based or expansion-based segmentation, we actually found that this was largely consistent at 88%. So this begged the question that whether we would get a similarly consistent result when we compare annotations between nuclear segmentation and cell segmentation. So the nuclei only contain about 40% of all transcripts. But despite this, the annotation, as you can see here, between the strategies is remarkably consistent at 87%. And you can see that when we get inconsistencies, these inconsistencies are largely between cell types that are quite related to each other. When we investigate where spatially these inconsistencies lie, then we find that they are in areas where the tissue quality was low and or there were low numbers of transcripts detected. So we were quite intrigued by this result. And so next we wanted to study how much representation we had of gene pairs that were mutually exclusive in single cell data in our different segmentation strategies. Sorry, my light just went out and I have to wave. Well, there we go. So, we first identified genes that were only co-occurring in about 1% of cells from publicly available data. And then on this plot here, you can see how often we find these gene pairs in either the nuclei versus the other two strategies. And you can see here, we always find these gene pairs occurring much less in the nuclei-based segmentation versus the cell segmentation, or even worse, the expansion-based segmentation, where this often occurs even more frequently. But let's go to a specific example of 2 marker genes. So in single cell data, these co-occur in only 2% of the cells together. But when we look at the nuclei co-occurrence, it's actually at 9%, whereas in the cells, it goes up to 23% versus the expansion-based segmentation, where it's at 33%. And you can really tell from this that, you're making your life quite a lot harder in identifying certain cell types when you're using expansion-based, but even cell segmentation versus the nuclei segmentation. So I now want to return to our pipeline. Even though I've just shown you that nuclei segmentation is actually quite great, and in most cases probably sufficient for what you want to do, let's assume you did undertake cell segmentation on your really high resolution confocal images. So typically we have around 400 images that are covering a slide, and they are overlapping slightly. So this requires us to stitch them back together to ensure that to ensure that you get like a complete image, but that also means that our cell segmentation masks need to be stitched back together. Unfortunately, at the time we were trying to do this, there was no tool for this. So we had to develop a really simple tool that can, in these areas of overlapping images, can just join cell segmentation masks together, split them, or remove cells completely depending on the context. And you can see what the tool does here. So once stitched, the next step is really to align the multiple modalities that we have. And in this case, the most important step is to get the transcript locations back to the cell masks. For all of this, we use ST-align, which can also provide alignment for slides that are quite a bit further apart. So we have also used this quite successfully for sequential sections of spatial transcriptomics data, as well as our spatial metabolomics data. And once all the sections are aligned and you have the transcript locations aligned to your cell mask, the final step is to determine just simply the gene transcripts that fall into those cell masks. If you want to give this pipeline a trial, then you can use this GitHub link here. Just a quick warning, this is all still very much under construction, but your feedback is super much appreciated. So we look forward to hearing from you. To provide some proof here that the alignment pipeline indeed works, I want to show you how our spatial metabolomics data actually matches up with our spatial transcriptomic data. So in the first image here, you can see the annotated spatial map with the different cell types. And then we made niches, spatial transcriptomic niches, where cells co-occur more frequently with each other on the right-hand side. And on the underneath that, you're actually looking at the spatial metabolomics data. And this is just a representative metabolite that you can see the spatial distribution of. And on the right-hand side of that, you can see the clusters, different metabolite clusters that have been mapped back to the cells. And what's really interesting is that these spatial transcriptomic niches that we are seeing are actually overlapping with what we're seeing on the metabolite data. And this led us to a really interesting discovery at the leading edge. So at the leading edge, we are finding upregulation of genes involved in choline metabolism, such as EGFR, which is shown here. And the corresponding leading edge metabolite cluster was also independently enriched for glycerolphosphocholin, which really confirmed the utilization of this choline pathway. So what was also really interesting is that when we looked at the immunofluorescence at this leading edge, we could see that there were actin-positive structures that were stretching across this area, which potentially suggests that choline metabolism is involved in the tumor infiltration here, which is possibly using these actin-positive structures. So to finish up, I just wanted to show you how you can use the 10x Xenium browser to really explore this type of data. And here you can see our immunofluorescence, and then we're overlaying it with some genes of interest, such as OLIC1, and then we can also look at EGFR. So even though you've done immunofluorescence outside of the 10x sort of ecosystem, it can all be brought back into the 10x ecosystem to really help you interrogate your data. So with that, I just want to finish up and say that I think in most cases, nuclei segmentation is really sufficient, and that should actually make your workflow a lot simpler. A lot of downstream analysis can be done in a manner that is actually segmentation agnostic or segmentation free. Immunofluorescence can really provide these intricate cell morphology information, but it does require a lot of computational work. So it's not always worth it, including stitching of confocal images, but we do have a pipeline for this now. And finally, that spatial multi-omics workflow can really give you consistent biological results that are across molecular levels, which is quite beautiful. And with that, I just want to thank everyone who was involved. It's quite a large lab, but really mainly Jürgen and Joel, who have really worked on this data quite extensively. Thank you.
Thank you, Saskia. That was really interesting. I already see there are some questions in the chat, which I will read in a moment. Feel free, that's a message to everyone, put your questions in the chat or raise your hand and you will be able to ask the question. So we have a few questions from Hussein. First one is, I think, oh, and Tuan also. So maybe I will start with Tuan, was the first question. Could I please ask what method did you use to cluster metabolomic data and how did you annotate metabolite from MZ?
So yeah, the annotation of metabolites from MZ values, I'll start with that, is actually like pretty hard, but we have a tool that is coming out soon called SPA-MTP, so watch out for that. That allows you to do this in a simpler manner, but I do acknowledge that this can actually be quite challenging. This is obviously not a metabolite talk, so I won't go much more into this apart from telling you to look into our look out for SPA-MTP when it comes out. It's mainly just matching the mass value with the mass value in annotations such as HMDB, but there is a little bit of work involved in that. Finally, for the method that we used in order to cluster the metabolomic data, this was actually quite simply done with first a PCA on the intensities, and then after that, just any sort of k-means clustering approach on the PCA.
All right, thank you. So I think we can move to Hussein's question. All right. Was immunofluorescence and H&E staining done on the same section as xenium?
So, as I said, it can be done on the same section as the xenium. You can absolutely do immunofluorescence afterwards, and then after you've acquired the immunofluorescence, it can actually then go to H&E as well, which is quite interesting. But so this wasn't done for all of the slides here, though, but we always do immunofluorescence straight after the xenium.
All right. What was the tool used for aligning sequential sections?
That's ST-Align, and yeah, we do recommend that because that's quite good and has worked really well and has a great interface to use.
How did your team find working with like the file outputs from the 10x software into ST-Align, and then you said back and forth kind of thing? How was that?
Yeah, I mean, there's a bit of a learning curve as with all of these things, but it's actually pretty seamless at this point. For us, it's more, the actual problems are the confocal images where there is a lot less sort of, I guess, information around on how you do that and how you get your confocal images stitched up. And that's why I talked about it so much.
Okay, good.
Yeah, so we had also a question, how did you choose the panels for xenium?
Yeah, I didn't actually want to talk about that because I think we got a bit lucky, I have to admit. So we started from a panel that 10x had actually designed for GBM research. And that was a great starting point of around 300 genes. And then we used lots of different papers that had done antibody-based work, but also single cell data in order to then really optimize our panel for our needs. But with 300 genes, it's kind of up to the beholder, I would say, on what you're going to end up putting in there. And I would also say you're always going to miss something. You do research. We've recently applied this technology in a clinical trial. And when we did that I really would have liked to have different genes turned out in the end on my panel, and I just didn't have them. But that's when, single cell data comes in handy that you might have from the same tissue, and you can then infer genes that you might not have measured. And that's obviously not as good as measuring them directly, but it gives you an indication of what might be going on.
So I actually have a question related to panels, because I think many people like we keep hearing, I want more genes. I want like all the genes. So 10x released the 5,000 plex panel. You were using the smaller panel. What's your take on needs? Like, do you think smaller panel is enough, or do you think more genes is better?
I mean, we would always like more genes, but I think working within your budget constraints, right? Like as you can tell from the stuff that we have done, even a 300 gene panel can give you beautiful results, and especially if you're matching it with some of 10x's single cell approaches.
Okay, yeah, thank you. We have quite a lot of questions.
Quite a few questions. Yeah.
We can take all of the questions unless Saskia, you need to go quickly somewhere.
So
oh no, just the last question from Hussein, which I see for now at least. Have you tried multicolor immunofluorescence using slide scanners to avoid stitching?
No, we haven't. But I think the stitching, you know, the nice thing about the confocal is that it gets such high resolution, which is what you really want.
Yeah.
So that's, and that, you know, yes, we could have downsampled as well and then stitched first and then done the cell segmentation later, but we do this on purpose on the really high resolution confocal images, because it does give us the best way to segment cells.
Yeah. And we have another question. Is there, from Paul, is there any shrinkage or similar changes between the different methods for the tissue section?
Yeah, I think so, especially when you're working with frozen materials, you get I'm not sure whether it's shrinkage so much, but just by when you're trying to put them onto the slide, so all of these technologies are slide based, so you use, I guess, with a fine paintbrush, you're trying to brush them on really carefully, and folding will unfortunately happen, and that is some of the largest pains that we have when we're trying to align slides back to each other with ST-Align, having to take that into account. But ST-Align is quite good at stretching things and, yeah, trying to shift it into the right position.
Also, actually related to sectioning, at the beginning I was wondering whether you did like separate, because you were putting multiple samples on your slides, right? So basically, and then with
I mean, I didn't talk about that, but we do that. Yes, we do.
You melted the FFPE and just joined it to have a nice one section, I assume.
Well, not quite, but we have previously, so we do typically try to get 2 sections on one slide just to be cost effective. Mm-hmm. And that is very, very much possible.
Yeah.
Okay, and G-Con had a message just about your paper, and they'd love to have access to it soon. So
yeah, we're working on it. It's like currently in first draft. Hopefully it will be out on bioRxiv really soon, but we're absolutely aiming for getting it on bioRxiv to get this message out that nuclei segmentation is probably sufficient for most cases, which I feel very strongly about.
Mm-hmm. Great.
And you have compliments from Klaus, great talk. Do you have observations on segmentation technology performance in cellular areas that are nucleus free, like axon bundles?
Yeah, no, I'm sorry. I know that. I mean, I would also love to study, you know, the processes of neurons. We've looked at, not many transcripts are actually outside cell boundaries first up, and then, you know, it's often unclear whether when you have a transcript that is outside of a cell boundary, whether it is because it might be a cell that is on top of it that you're not actually quite capturing, that was cut off where the nucleus wasn't there anymore, but you still have part of the cell. Yeah. And those are the ones those are the transcripts that you're seeing, or whether you're truly seeing longer sort of transcripts that are accumulating in, you know, processes of neurons. So unclear whether you can actually do this, and we are certainly not the lab that is investigating that.
And then Dina also said, thank you for the great talk. And why did you choose ST-Align instead of the native Xenium alignment tool?
I think the native Xenium alignment tool didn't exist when we started. That was number one.
I think you were too early. Yeah.
So we were a bit too early for that. And I guess it comes back for us to wanting to have it all in a computational pipeline at this point. So it's, you know, it's all done in Python. It can be batched. It can be bundled across loads of different samples with, you know, and it's not super hands-on. That's been for us the important bit.
And is it spelled ST-Align or is it?
Yeah, it's spelled ST-Align. I should have, and I'm sorry I didn't put the paper there. That is a super big oversight. But if you Google it, I'm sure you'll find it.
All right. So we are just on
minute.
time, but you have 5 seconds to put your last question to join this conversation. Otherwise, we are hoping to see you also next time. One last question. Okay.
Oh, no, just
no, just a comment. Yes.
Yeah.
Yeah. So thank you, everyone, for joining us today. Thank you, Saskia, for your presentation. It was really interesting. And I hope I will see you, everyone, during our next spatial conversations. So please stay tuned. You will receive information about the next one.
Yeah, and thanks everyone for your questions today. It was really great to have lots of questions, so keep them coming. And even if you want to be unmuted next time, we can unmute you and you can ask your questions yourself. So
yeah, it's spatial conversation, so let's have a conversation.
Yeah, great. Thank you, everyone. Thanks, Saskia.
Thank you. Bye.
Bye.